Today we're going to be reviewing the protein collection and testing. We have a couple objectives that we want to make sure we can outline. We want to make sure we can outline the steps from idea of a protein to sale. This includes testing and sip dips. We want to be able to discuss assay types. And lastly, summarize lab skills used to accomplish this. And some of those would be things like the zone of inhibition and making solutions. This unit can be chunked into three major ideas. The first being, how do we come up with products? So the first step is that we can idea a product, and we use things like zone of inhibition. We can look at rainforests and natural treatments. From there, how do we go about testing for its usefulness? How do we go about determining if it's marketable? And how do we go about checking its quality and use with the FDA? So these are all the ideas of sip dip and the questions that you need to be able to ask. Secondly, we'll look at assays. So one of the big ideas is that there are multiple types of assays. But the main differences that we're looking at is that there's a presence assay. How do you know if something's there? Versus an activity assay, which tests a lot of times for an enzyme. This would be an example. This would be the amylase. We're looking at a color change to determine if something's happened. And lastly, one of the major big ideas from this unit is purification and testing. So the purification was all about the sorting, using things like column chromatography, and we also talked about antibodies. And the testing, which relates back up to the FDA quality control. How do you know what you're doing is actually effective and it's not a placebo effect? So those are the three big ideas of this unit. You also wanted to focus on what were the main labs of this unit. So the main labs of this unit had to do with antibactericides and antimicrobials. This had a lot to do with zones of inhibition and how you measure those. So if this is your plate and you have a lawn, you'd want to measure this area that's created by the antibactericide. We also talked about presence assays. So this, for the very first time, this was kind of our starch with iodide and Benedict's. The activity assay was where we had amylase. We were testing for the presence of an enzyme. So the difference between these is presence just tell you if it's positive or negative. It's there or it's not. Activity assays can tell you kind of how much, and this was determined by kind of the percentage or the amount of glucose that was present. Lastly, our last lab for this unit had to do with the standard curve, which was also a presence assay, but it had to do also with an unknown that you were trying to calculate. So this was the idea that you could extrapolate data using a curve, we did caffeine in our case, but using this curve, once you've kind of created it, if you know any unknown points, you can then make estimates and predict concentrations or the amount of a substance in a certain area. In terms of skills that you need to know for this unit, I talked a little bit earlier about measuring the zone of inhibition in terms of bacteria, calculating chi-square, and this was just basically having to do with kind of lab effectiveness, using a presence assay. So this was the whole thing with starch and iodide. Analyzing results with controls, both positive and negative. How do you know if your results are valid and reliable? Using a spectrometer, basically what are the steps? What would you need to do that? And using the standard curve to determine the unknown. This is going back to extrapolating data. So knowing that this is the curve, can you pick a point out here to understand? Can you pick a point down here to estimate? Those would be the important skills from this unit. In terms of vocabulary, here's kind of some big vocabulary that I picked out. Feel free to pause, look up, and pick kind of things that you might not know or know of. So to kind of wrap up, what was the review of this whole unit? You wanted to know how to make medical treatments, how they're identified, developed, tested, and sold. This had to do with kind of the marketing, the sip dip, those ideas, the ideas of purification. You also wanted to be able to use math and scientific reasoning to analyze results on testing effectiveness. In terms of study, my recommendations are for you to Review labs, the lectures, and quizzes. You should have gotten all this stuff back. If not, please make sure you get it back before the test. And my last little tip to you is to please make sure you take some time to review, write some questions that you might think show up. This the same idea of making a cheat sheet. It really helps you a lot to process and condense information. So that would be my last recommendations for you at the end of this lecture. And good luck reviewing for this unit. I wish you a lot of luck on the test and exam. If you have any questions, please feel free to call or email me. And then we'll get going on our next topic. Next week, we'll get to talk about plants. We'll be talking about plant biology. So keep that in mind, thinking about what you want to learn and what you don't know about plants. Hope you have a great one. Take care and have a great night.